Now that we have looked at the morphology of the sperm and the egg, we'll look more closely at the adaptations of these amazing cells that allow them to meet each other and to form a new organism. And in this lecture, we're going to consider the events that happen in the fertilization of invertebrates, using the sea urchin as a model organism. And in the next lecture, we'll discuss mammalian fertilization. In the sea urchin, chemoattractants activate and guide the sperm toward the egg jelly. Once the sperm reaches the egg and makes contact with the egg jelly, the acrosomal reaction is triggered. This results in the release of digestive enzymes from the acrosomal vesicle. In addition to the release of these enzymes, globular actin that is located under the acrosomal vesicle polymerizes into filaments, and it forms the acrosomal process. And here's what the polymerization of globular actin into filamentous actin looks like. The acrosomal process and the enzymes that were released from the acrosomal vesicle help the sperm to digest a path through the egg jelly, and eventually the sperm binds to the vitelline envelope. This is then followed by the fusion of the sperm and the egg plasma membrane. Because fertilization in sea urchin is external and the gametes are released into an aqueous environment that is very large as an entire sea, the gametes become very diluted, and that decreases the probability of successful fertilization. An adaptation to this is to release a very large number of gametes. Take a look. Another problem associated with external fertilization is that the gametes are shared with the gametes of other species. So there has to be a mechanism to attract and to activate the sperm to allow the binding of the egg and the sperm in a species-specific way. The attraction of the sperm to the egg in sea urchin is accomplished by sperm-activating peptides, and these are chemoattractants that are made by the egg and they diffuse from the egg jelly. The saps are secreted by the oocytes once they are mature, so once they're able to be fertilized, and they attract the sperm in a species-specific way. In sea urchin, the sperm are actually immotile. They're not able to move when they are inside of the gonad. When the sperm are released from the gonad, there's a change in pH from more acidic to more basic, and that allows the sperm to gain motility. That change in pH results in the activation of the motor protein dyning, which we talked about earlier, which is located in the axoneme of the flagellum. Although the sea urchin sperm gain motility when they're released from the gonad, that doesn't mean that they have direction. That doesn't mean they know where they're going. So the saps play a role in helping the sperm reach the egg. These sperm activating peptides or saps bind to receptors on the membrane of the sperm, and the name of this particular sap is Resact. The names vary depending on the species. The binding of the sperm activating peptide to its receptor leads to the opening of calcium channels, and these calcium channels are known as cat spurs. As a result of the opening of these cat spur channels, calcium flows into the cell, and that leads to an increase in the synthesis of ATP in the mitochondria and the activation of the motor protein dyning, which is located in the flagella. So this actually helps to improve the motility of the sperm further. In addition to inducing changes to dyning, which promotes sperm motility, saps also serve as chemoattractants for sperm, as I mentioned previously. If sea urchin sperm are placed in petri dishes, they swim aimlessly without direction. They're basically going around in circles. But as soon as the sperm activating peptide or sap is added to the petri dish, the sperm begin to migrate toward it. So the sperm migrate following the concentration gradient of these peptides. By following the sap gradient, a sperm is eventually able to reach the egg and to make contact with the egg jelly. As I mentioned previously, contact with the egg jelly triggers the acrosome reaction. This reaction results in the formation of the acrosomal process, and it also involves the exocytosis of the acrosomal vesicle. And to give you an idea, here's what exocytosis looks like. But what is it about the egg jelly that triggers that acrosomal reaction? Well, within the egg jelly, there are sulfate-containing polysaccharides. And these are actually species specific. So different species secrete different sulfated polysaccharides, which bind to their corresponding receptors on the sperm of the same species of sea urchin. When these sulfated polysaccharides bind to the receptors on the sperm cell membrane, they trigger the exocytosis of the acrosomal vesicle and the formation of the acrosomal process. The fact that these saps are species-specific has been shown by experiments. In one of these experiments, sperm from different species of sea urchin were incubated with sulfated polysaccharides from the three species. And what was found is that only the sulfated polysaccharide from the same species of the sperm was able to trigger the acrosomal reaction. So in the y-axis here, we see the percent of sperm that have undergone the acrosomal reaction. And on the x-axis, we see the three different species, A. lixula, E. leucanter, and L. variegatus. What we see is that when the sulfated polysaccharide from A. lixula is added to the sperm from the same species, that triggers the acrosomal reaction in the sperm. But if the sperm from A. lixula is incubated with sulfated polysaccharides from the other species, the E. leucanter or the L. variegatus, we see that those do not trigger the acrosomal reaction. This is important because it helps to prevent interspecies fertilization. The sulfated polysaccharides trigger exocytosis of the acrosomal vesicle because their binding to their receptors leads to changes in calcium concentration in the sperm cytoplasm. The activation of the receptor leads to the opening of different transport proteins in the plasma membrane and to the activation of different enzymes. But rather than focus on the names of the proteins, I want you to pay attention to the overall effect of these polysaccharides, which is to increase the calcium concentration in the cytoplasm of the sperm. This increase in calcium triggers the exocytosis of the acrosomal vesicle. And the reason I want you to pay attention to this particular detail is this. Calcium is the common trigger for exocytosis. This is not just true of the sperm, but is also true of other cells as well. Another important thing to note about exocytosis is that when it happens, the membrane of the vesicle that's fusing with the plasma membrane becomes part of the plasma membrane. So in sea urchin, the membrane of the acrosomal vesicle has a protein binding, which is necessary for the sperm to attach to the egg. So this membrane protein is originally inside the sperm, it's not exposed. Once the acrosomal vesicle undergoes exocytosis, once it fuses with the plasma membrane, this membrane of the acrosomal vesicle is going to become part of the plasma membrane. So the binding proteins are now going to be located at the plasma membrane of the sperm. The protein binding has a great name because it's important for the binding of the sperm to the egg, specifically to the vitelline envelope of the egg. It also helps to promote the fusion of the sperm and egg. This protein, like the sap and the sulfated polysaccharides, is also species-specific. In an experiment, binding from two different species, S. purpuratus and S. franciscanus, was incubated with the jellied eggs from these two species. And the eggs are the larger circles here, the binding particles are the smaller circles. In this diagram, the colors represent the species, with the purple appropriately representing the S. purpuratus, and the green representing the S. franciscanus. As you can see, when binding from S. purpuratus is incubated with eggs from the same species, that causes the agglutination of the eggs because the binding binds to the eggs. However, the S. purpuratus binding does not cause the agglutination of the S. franciscanus eggs, and that shows that the binding does not bind to the eggs of this species. So binding is the third mechanism used in the sea urchin to prevent interspecies fertilization. And the existence of these multiple mechanisms to prevent interspecies fertilization is common for species that have external fertilization because the gametes of different species all share a common habitat. These mechanisms are important for preventing the fusion of germ cells from two different species, which would result in an unviable offspring. The protein binding not only helps the sperm to bind to the egg, it also helps to promote the fusion of the sperm and the egg cell membranes. So it's a fusogenic protein. As the sperm begins to fuse with the egg, actin polymerization in the egg leads to the formation of a fertilization cone. So we see that actin polymerization is important for fertilization. We see it in the formation of the fertilization cone, the microvilli in the egg, and also the acrosomal process in the sperm.